targeted compounds such as small molecules, peptides, and proteins to carry a radioactive payload to a tumor while minimizing damage to healthy organs. The choice of isotope selected allows one to combine diagnostic images with targeted radiotherapy, either in the same agent or using different agents targeting the same tumor antigen. I'm delighted to be joined today by four key experts. First up, Dr. Deborah Charych, co-founder and chief technology officer at RayzeBio, an oncology company focused on targeted delivery of radionuclides. Dr. John Valen is up next, founder and chief executive officer at Fusion Pharmaceuticals, a clinical stage biopharmaceutical company developing next-gen radiopharmaceuticals as precision medicines for the treatment of cancer. Next up, Dr. Guillermo Garicki, Chief Medical Officer of Advanced Accelerator Applications at Novartis, an innovative medicines company focused on the development of products for targeted radioligand therapy and precision radioligand imaging. Lastly, I have Dr. Ohad Ilovich, who is the Senior Director of Translational Research at Invicro, a Konica Minolta company, a global provider of imaging biomarkers, theragnostics development services, clinical imaging core lab services, and advanced analytics for drug discovery and development. So we have about an hour for today's discussion, and we will be taking audience questions towards the end. So as soon as you think of that question, hit the Q&A button at the bottom of your screen, and I will pose that to the panel towards the end. So we're breaking today's discussion into three sections. The first section is target identification and early development. Then we're going to move on to translational and clinical development. And lastly, we'll discuss what the future holds. So let's start with section one. And I'm going to actually kick this off with John here. John, could you weigh in on the resurgence of theragnostics in the recent years? What's driving this? Is it isotope availability or platform availability, or the success of a handful of agents?
Great, and thank you for the question and for the opportunity to speak today. So having been in the field for about 25 years and had the great fortune to do radiopharmaceutical manufacturing for a portion of that, I've certainly seen firsthand how patients get tremendous benefit from diagnostic and therapeutic radiopharmaceuticals. And I'm really pleased to see the growing recognition of the potential value of this field. But I'd say, like many areas within oncology drug development, it takes time to advance. There are technical hurdles, there are commercial hurdles that have to be overcome. And, we've seen this in other areas, ADCs, immuno-oncology, gene therapies. So once you overcome those technical and commercial challenges, you start to build momentum. And certainly, I think in the radiopharmaceutical field, the big driver is data. And we've seen some fantastic clinical data emerge, going back to Xofigo, Lutathera, Azedra, and then the great data that came out about PSMA, really enables people to see the potential within the field. If we look back historically a little bit in the field, a big driver was a lot of the studies were done at academic institutions where they did phenomenal work developing the technology, getting initial patient data. But there was a big emphasis on diagnostics and a little bit more recently on therapeutics, and they were small numbers of patients. When you combine that now with the investment from biotech and pharma, with the ability to do larger, more powered, randomized clinical trials, the data and the potential value, I think, is becoming much more apparent. Another driver for me, and I think this goes back to the timing component, is how the clinical practices are changing and evolving towards using multidisciplinary teams. And obviously for nuclear medicine type techniques, you need expertise that spans medical oncology, radiation oncology, nuclear medicine, radiology, nursing specialists. And so we have I think the collective knowledge and how we manage patients that can leverage the potential from the field. I think the other thing that is driving this momentum is, if you look at some of the existing agents, the Lutatheras, PSMAs, and others, people are seeing the value of leveraging the simple mechanism of action there's an enormous untapped druggable space within that radiopharmaceuticals can go after. And I think people are now recognizing how they can leverage that mechanism of action. So from my perspective, we're scratching the surface. We're seeing data drive the field, the users being excited about the technology, the ability to use imaging to select patients. And I think this momentum is really going to grow and continue.
Right. Anyone else here on the resurgence of theragnostics here?
Maybe just one facet to add. I very much agree with what John just said, and that's the safety perception. We're always up against concerns about radiation safety. And as we now see well-conducted studies present long-term safety data, like for Lutathera, I think that eases many skeptics that we don't have a high rate of secondary malignancies, and that provides confidence to physicians and patients to use these drugs.
Now, I'll just add one more note, on the efficacy side. Since we just heard about the safety side, thank you, Guillermo, for that. On the efficacy side, being in cancer research for as long as I have been, we like things that work. And when something works, it really, one starts to pay attention. And I think the efficacy data that's been coming out over the past few years has really also driven the resurgence in this field.
Very good. Very good. Let's talk about the development of theragnostics and some of the platforms. Mostly it's been small molecules, peptides, and antibodies. But maybe, Guillermo, you could kick us off over here. Are there some advantages or disadvantages you see with each of these platforms here to develop theragnostics?
Yeah, thank you, Arslan, and thanks for inviting me here to this great panel. We have horses for courses, I would say. We have small molecules, we have peptides, we have antibodies or small biologics. And I think in long term, we'll probably see a mix of those. They all have advantages and disadvantages. Obviously, the high affinity and great availability of biologics with high specificity is a great starting point for development of radioligands. And John already mentioned the repurposing of ADCs and other technologies that have been proven to be successful just by changing the warhead, if you will. On the other side, small molecules have a preference in terms of permeability and pharmacokinetic properties, in vivo stability, and manufacturing scalability. So I think there's no right or wrong. I hope that we're going to see a lot of research along all these platforms and do the best we can for patients with a good mix.
Okay. Deborah, do you have anything to add here on the platforms?
No, I think that was well said. The main component I would add is that we've focused on pharmacology and pharmacokinetics. So, we think we have an understanding of what kind of pharmacokinetic profile might be good for radiopharmaceuticals. So we try to drive towards those types of platforms.
Okay. Maybe I can just add one quick point. Please. So in our field over decades, people have fallen into certain camps of liking certain classes of molecules or certain isotopes. And I think one of the things that you're seeing evolve in the field is data. Data is driving people towards pick the right tool for the right job. So I completely agree with Guillermo and Deborah that really it's about identifying the right molecule, the right target, the right isotope, and let the data drive forward as opposed to sort of feelings about particular classes. So I think we're going to have exactly a diverse array of radiopharmaceuticals to choose from.
Okay, wonderful. So we've seen significant success targeting tumor-associated antigens like PSMA and somatostatin, and some increasing interest in using a tumor microenvironment antigen like FAP. So which antigen strategies could lead to the development of both novel and successful theragnostics? Deborah?
Yeah, so again, coming from the cancer biology and cancer research side, I guess the way we've been thinking about it is really from the tumor target point of view. Like getting to first base is really, finding a really good tumor-restricted target that's on the cell surface, ideally. Whether such an ideal target actually exists, may not. No target is perfect. But I think if we start with the tumor biology first, and then work hard to find good chemical matter to target those targets, then I think that's a reasonable strategy. Sometimes there may be cases where the chemical matter exists, because there's already a peptide or something like that. But the target may not be the best tumor target necessarily. So I think working backwards from the clinic, from the antibody drug conjugate space or the CAR T space, there's probably no shortage of good tumor targets. The getting to second base and third base is really about finding the good chemical matter to target those targets. So that's the kind of the philosophy that we've taken. Stromal targets are interesting. I think something like FAP clearly has benefit in the imaging space. My impression, and this is just my impression, is that the jury may still be out on the therapeutic benefit because there could be potentially complex mechanisms of action using a stromal target. But from the imaging modality point of view, that there's no question that FAP seems to be potentially a winner.
Okay.
Yeah, and I think it's very interesting that for a lot of the standard toxin delivery platforms, targeting something like FAP, where the agent will have to then diffuse and then enter the nearby cancer cells and destroy them, that's really not a blockade. It's not an obstacle for a radiotherapeutic like a beta emitter, because even if it attaches to one type of cells, it'll have a radius around it of the cells that are getting affected. So I think the combination of the theragnostics and the taking advantage of the tumor microenvironment targets is really interesting.
Very good.
Yeah, I think one other thing I would add is that I think this highlights the broad potential of the radiopharmaceutical space. Traditionally, we've all focused on a limited number of targets, but when you look at that mechanism of action, the ability, and I think Deborah pointed this out, there are lots of tumor antigens that could be very highly attractive for going after with a radionuclide approach. So I think the ability to develop innovative therapies in areas of high unmet need, there's a tremendous amount of potential.
Great. Excellent. I see audience questions coming in right now, so I want to thank the audience for that. We are going to get to questions towards the end of the session, so keep them coming in. Let's move on to our second section over here. So we have been talking about target identification and early development, but let's move to translational and clinical development now. Ohad, here, I'm going to shoot this question over to you here, but how does the translational development of a theranostic differ from that of a regular therapeutic?
It's a great question. So I think there's many small differences between the different drug development pathways, but there's a few that highlight major differences that I think people need to grasp before they start. The first one is chemistry. So when you're working with the radioactive isotopes, there's a few new dimensions that have to be taken into account, like specific activity, which is the ratio between the radioactivity that's loaded onto a molecule and how many molecules are associated with it, the physical half-life of the decay of the isotope, which puts limits on, for example, you can't just make the agent and then store it for 6 months and dose a patient. Usually you have to prepare it very close to dosing. And things like mass differences where you can't just dose a lot of antibody or a lot of peptide into patients. And really in standard therapeutics, who would even think of dosing a patient with 100 micrograms of peptide to get a therapeutic effect? Beyond the chemistry, there's biodistribution. So taking an example of an ADC, there's a lot of work that goes into developing an ADC on the in vitro space, making sure that the mechanism makes sense, that it's killing the tumor cells and so forth. Then when you move into in vivo, there is some PK investigations that go into effect, but it's mostly a pharmacodynamic type testing. So making sure that the ADC is actually killing tumors, but is not damaging the normal organs. With a radio-labeled antibody, for example, there's a lot of imaging that goes into drug development to figure out where is it going. Is it accumulating in the spleen? Is there a pathway of excretion through the liver? Is it excreting through the kidneys? Is it retaining in the kidneys? One thing you have to remember is that, for example, an ADC that's floating around in the blood and not attaching to any kind of cells because none of them have any antigen and it's stable, it really has very minor side effects, but a radio-labeled antibody will have radioactive decay all the time. So even if it's just floating around in the blood, it is going to irradiate the blood, the bone marrow, the organs that it's entering. And that sort of leads us to the third major difference, which I think is safety. Again, with an ADC, for example, you would have to take care of the safety of the protein, the antibody itself, and then the drug conjugates, make sure it's stable, they're not destroying normal organs. But then when you're doing it with a radio-labeled antibody, you have to take that extra dimension of radioactivity and see where is the isotope accumulating, whether it's still connected or unconnected to the antibody. And that gets taken care of by dosimetry, basically figuring out the accumulation and distribution of the isotope. And that dosimetry profile, which goes on top of the tox profile in the safety package, is investigated both non-clinically and clinically.
Wonderful. Anything? That was a very comprehensive thing. Yeah, Guillermo?
Yeah, maybe just to look at the positive side, right? I mean, Ohad explained quite a number of challenges that we have with the radioligands. Having been in drug development oncology for many years, actually, I love these radionuclides because what you see is what you treat. You see where they're going. You have the perfect biodistribution. And assuming that the majority of the effect, it really is a local effect of where you get the radiation, you can de-risk your development in phase 1 so much compared to other drug developments that it's really fun to do these early clinical studies and really get close to the patients and see what you get.
I couldn't agree more, Guillermo. You know, from a drug development point of view, it's fascinating because you can get PK in the blood, PK in the tumor, PK in the kidney, PK in the liver. You get PK. So it's really about pharmacokinetics and so many varieties of tissues all in one fell swoop, which is, you know, impossible to do, especially in the human, with regular drug development. So that's really quite attractive. And then on the pharmacology side, being able to select patients who are actually expressing high levels of your target, you can do that by imaging without having to take biopsies, you know, which is huge advantage, I think, and it really will help move oncology forward. So the non-invasive component of being able to select patients, I think, is a huge benefit.
Yeah, I agree. And I would say that one of the other components that we should never forget about, we're talking about the differences in development, is also the dosing schedule and the idea about total cumulative radiation dose versus, you know, the sort of steady state concentration of the actual drug itself, which differs from other therapies. So a lot of these factors we have to take into account. But I mean, the ability to select patients, measure PK, it's a tremendous opportunity. And also tune the isotope to achieve the properties that you're looking for. Very unique part of this field.
All right. Well, let's pivot into actually the path into doing first in human studies in theragnostics. So here's my question, Guillermo. I'm going to pose it to you, but really, here's the question. So, are we running consecutive imaging and therapy studies, or versus a combination imaging and therapy study?
Yeah, so I think ultimately, as Deborah just pointed out, we'll need the radioligand imaging to select optimally target these patients. So I don't think they will go away as a diagnostic and screening tool. And even beyond theragnostics, many of these radioligand imaging tracers will provide great value in oncology by steering patients to optimal treatment paradigms. In development, ideally, you have a pair of isotopes that can be attached to the same ligand and chelator, like gallium and lutetium. And then it's actually not a lot of work to have the imaging first and then do the therapy back to back, if you will. That is kind of the ideal situation. Now, with some of the ligands, with some of the isotopes, that's not so easy. But as Deborah said, the imaging provides so much value about the biodistribution, about the pharmacokinetics, that I don't think anybody would go kind of blind into, let's say, an alpha therapy, where you can't do biodistribution on the therapeutic isotope without also studying the imaging. So whether it's in two separate studies or in the same combined study, I see them as theranostic pairs, and that's why we call this theragnostics, and we are quite excited about it.
And I think one thing to add is that if you're doing an imaging only study, then you're basically asking patients to withhold on therapy for be it a week or be it a few days. And that's a difficult sell, and that's really unfair to them. I think if you're running a study that has the imaging component and then the therapeutic component, and you're planning the therapy dose escalation component in a way that makes sense, then these same patients that are getting the imaging agent can also get a therapeutic benefit, which makes it a lot fairer to them, and it makes patient recruitment much easier and much faster, so you can get the clinical results you're after much quicker.
Great. I just want to keep thanking the audience. We're getting a lot of questions in, some really nice questions over here. So I want to keep the panel going along with our discussion, because I think it's getting some great questions in from the audience as you guys are discussing over here. So the compassionate use model that is prevalent in Germany, is it a desirable path for proof of concept to make a go or no-go decision, or is the lack of standardization make it unattractive? Guillermo, do you have an opinion on this?
Yeah, I think it's a bit of a trick question. There are several European countries that have regulation that allows compassionate use without a full protocol and things like that. And that has been quite useful in the academic discovery and development of radioligands. What I see is a convergence of safety and quality standards between compassionate use and proper clinical trials. So I expect that in the future we'll see some of this still be in the academic space, and it will contribute to innovation. But eventually, agencies will ask for properly conducted clinical trials, for a good dose escalation, for documentation of clinical trial data. So it will never fully replace the clinical development that we have to deliver.
Okay. Any other opinions on that one real quick before I move on to our next question here? Okay, so currently the accepted limits on kidney and marrow doses sit at 23 and 2 grays, respectively. Those limits originate mostly from external beam radiotherapy and may not be appropriate for alpha beta therapy. Do you have any thoughts on that, Deborah?
Yeah, so that's somewhat of a hot topic. Excuse the pun. But if you dig back into the literature, the 23 gray, if I read it correctly, was set like in 1952. And that was using X-rays, and X-rays have a fairly broad reach. And so it was a very defined study that was done, and the 23 gray emerged. And I think back then the units were Roentgens. I can't even pronounce it. But I do think that is probably too conservative. That could be just my opinion, but I think the 23 gray is probably on the low end when it comes to systemic radiation, which is what we're doing. So I think there's a lot of upside there potentially. And there are some small studies like out of Sloan Kettering and other academic centers, which show that you can actually provide, greater radiation dose to the kidney without necessarily having kidney protectance or without any undue harm to the patients. So I think the jury's still out. There aren't any controlled clinical studies, like large studies that would help necessarily change that limit at this time. But I think it's going to evolve because it does feel like that's probably on the low end. We could probably go higher.
Any other opinions on this one over here from the panel?
I think maybe my comment is that it's a guide, and now we have products which are going in large numbers of patients, and we're looking out numbers of years. So it's going to be clinical data, and I think Guillermo mentioned this earlier about well-controlled safety studies, looking out long-term, that we can then go back and re-look at those values, which I agree are historic. But the nice part about radiation, it's fairly well characterized, but we'll use clinical data to set what the real limit should be when you look at the different constructs. So this is definitely, in my opinion, going to evolve with time.
Yeah. And one thing, if I may add, I totally agree, is the type of radiation may matter as well. Like that 1952 study was X-rays, which go a long distance. If we're talking about beta particles or alpha particles, those are increasingly of shorter and shorter path length. So the potential for damage is less to normal tissues. So I think the type of radiation may be important as well as the absorbed dose.
Excellent. All right. Now let's go ahead and move on to our last section over here. Again, to the audience, thank you for your questions. We're going to be getting to those in about 10 minutes or so. There's a lot of questions in, and so don't be shy. Please get them in. We're going to ask the panel some really great ones over here. But our last section here, what does the future hold for theragnostics? So what's the future beyond the use as a single agent? Combination therapies with DNA damage repair inhibitors like PARP-I, checkpoint inhibitors, other immunomodulators? How do you plan for combinations that would make sense? How about John Valiant?
Sure. So, I think you've seen the potency of radiopharmaceutical monotherapy being nicely demonstrated. But if you look at the mechanism of action, there's really an opportunity to move radiopharmaceuticals from later to earlier line therapies and to expand their utilization by leveraging that mechanism of action and our increasing understanding. So certainly in our case, we're very interested in looking at combining radiopharmaceuticals with DNA damage response inhibitors. I think that's a natural sort of association between the two, where the radiopharmaceutical brings the DNA damage, the DDRI prevents the repair. And really this is mutual benefit, in the sense that the DDRI typically work in specific patient populations where there's the appropriate mutation, where the radiopharmaceutical can now allow it to work in wild type. And we reported some data at ACR showing that we could take a no effect dose of the radiopharmaceutical in a PARP insensitive model and really start to see synergy between the two types. The challenge, of course, is looking at overlapping toxicities and how you manage that. But certainly, I think that is one area that really has tremendous potential, both for single-strand breaks as well as double-strand DNA break repair. And then the second one, I think really is immuno-oncology. Early days, external beam showed that distal metastases not directly irradiated would show some response, but it was really more anecdotal, not well-controlled studies. But now people are doing larger studies looking at combination with checkpoints. We certainly have seen a mechanistic rationale with alpha therapies combined with checkpoint inhibitors to really drive tumor neoantigen formation. So really, I think there's an opportunity with these sort of innovative earlier line therapies to leverage what we're seeing with radiopharmaceuticals.
Any other comments from the panel? All right. Okay, so more on the future here. What techniques make the most sense to spare dose to normal organs and expand the total dose injected in patients? Is it pretargeting, radioprotectors, blocking antigen in normal organs? Is it PK retention, manipulation? Ohad, how about let's let you pontificate here.
Yeah, I think that's a really interesting question because I think we could probably have spent half the roundtable just talking about this, but I think the most commonly used method for radiolabeled peptides, for example, has been a co-infusion of a peptidic solution that's meant to overwhelm the polypeptide reabsorption system in the kidneys. It's basically coating the inside of the kidneys to make it simplified and avoiding the reabsorption of the radiolabeled peptides back into the kidneys and its retention in the kidneys. So that's been used often and for years upon years in the clinic. Because that strategy isn't really viable for proteins that have long circulation half-lives, you can't really expect a patient to have an IV in his arm and get infused with peptides for 3, 5, 10 days. There's been a lot of interesting work being published lately where you use PEG spacers between the proteins and the radiolabels to induce the formation of short-lived non-retained metabolites that have improved clearance profiles. And that basically, instead of the radiolabeled antibody, for example, circulating for a long time, it enables it to, and then retain the normal organs. It enables these metabolites to wash away quickly. So then when they do accumulate in liver, for example, or spleen, they wash away quickly, and then the dosimetry profile is still very much acceptable. And I think one more thing that I've seen recently, it was always, so pretargeting was always a complex and interesting platform. It has been developed in academic situations. I've seen it a lot. But recently, so first of all, what pretargeting is basically you would inject a long circulating antibody, for example, or a biologic of some sort, let it decorate a tumor cell, and then after it clears from the blood, you give it a few days and you follow up with a very short-lived molecule that's carrying the isotope. And that isotope adheres to the antibodies that are, or the biologic that's adhere to the tumor cells. And then you spare the normal organs from basically having the antibody circulate for a long time, irradiating them. It's a very flexible system, and it has a lot of variables. So if you think about it, when you develop something like this in the clinic, then how much dose, how much protein do you give in the first agent? How much time do you let it clear? When you follow up with the radioactivity, how much activity and how much of the actual mass of the molecule do you need to give as a follow-up? And it's complicated, but I think the advantages, if you do manage to figure it out, are really high. And I've been really happy to see that a few of the industry companies have taken it upon themselves to try these out in the clinic. So I think these are really interesting pathways to spare normal organs.
Any other discussion on that from the panel here?
Yeah, I mean, I would love to chime in, and thanks so hard for that. You know, we've taken, speaking about pre-targeting, we've actually not done that approach, but we've taken a more, I guess, traditional drug development principles approach, to sparing normal organs. And so first, we want to start with really good tumor targets. So if the target is tumor restricted, that already may go a long way towards sparing normal tissues. The second approach we've taken is to apply good pharmacology and good pharmacokinetic principles, to target the target so we get that rapid clearance everywhere else, except where in the tumor where we want it. And then the third approach is to use an isotope that has a short path length. So something like actinium-225, which has a short linear energy transfer range, that may produce the most sparing of normal tissues. So those three principles we thought may provide a potentially simpler approach to sparing normal tissues without having to invoke multiple agents in development or multiple combinations of strategies. So we just wanted to keep it simple if we could.
Great. All right. I think it's about time to now move to the audience questions. And so there's still time to get them in. And if you guys have any more questions as you think of them, please hit the Q&A button at the bottom of your screen and get those in over here. Let's start with a question from Wendy in the audience. So Wendy asks, do you need to validate the imaging agent for patient selection in a clinical trial with a biopsy in the initial development? Who would like to take that? Ohad?
Sure. So I think if you're, again, so Guillermo described that there are basically two ways of developing these theranostic pairs. Sometimes it's the same molecule and you simply switch the isotope. And the other one is using completely different agents. I think if you're using completely different agents, it will definitely make sense that whatever you're using for imaging, you make sure that it's really imaging the target. So doing probably a phase 0 or more study, looking at the imaging characteristics and the antigen expression in the tumors makes a lot of sense. I think you get more flexibility if it's the same molecule with two different isotopes, because then if you're able to show bioequivalency to the FDA, I think you're basically sort of jumping over that first need to show that it's really very specific, because the imaging agent is more giving you a profile for the therapeutic that's going to follow it. And I think if you then, in an initial phase, if you can show that you're selecting patients according to the performance of the imaging agent, it clears the path a little more easily, and you don't have to really jump through a lot of hoops to show that it's very specific to the antigen and do a lot of validating.
Okay. Anyone else want to chime in on Wendy's question here?
Yeah, I think maybe it's a great question, and we have to acknowledge that the whole theranostic space is still an emerging platform, also with the regulatory authorities. And we're working very closely with the FDA and other agencies to see how we can streamline the development. And as we discussed, I mean, there are the standalone imaging agents, and then there are the strictly patient selection imaging agents, and there may be different paths evolving in the future. So it's a very fascinating area, also from a design of clinical development and regulatory interface perspective.
Yeah, and I'll pick it up. I totally agree with what Guillermo's saying. It's really a novel field. And I think if you look at the FDA as your friend, and you first of all, before you do anything, get some advice, go for the pre-IND conversation, figure out what it is that they're going to let you do. And then following up and taking that advice and following up on it, I think it's really the path of least resistance, trying to cut corners or trying to, reduce imaging time points in patients too quickly to make life easier or to make the recruitment easier. I think it's going to be a difficult path if you try that.
All right. I have a question here from the same anonymous. This is a very direct question. Do we currently have enough actinium-225 to support clinical trial demand? It's a very direct question. Ohad, do you want to answer?
Ooh, that's a doozy. So with the actinium, you have to take into account that there's basically two production pathways that are commonly used. One is when you're milking a generator. So basically the thorium is coming out and it's very clean and it's pure, and most of the people are using this in the clinic. When it's cyclotron-produced actinium-225, the current production methods that are common have it being produced with a very minor contaminant. It's actinium-227, but it's very long-lived, and it's also an alpha emitter. So it's a lot of companies that we've been working with right now have been trying to avoid using the actinium-227 contaminated compounds, because again, it's another layer of complexity, and it's a long-lived isotope, so that complexity is really difficult to figure out in a short amount of time. If you're only using the pure actinium that doesn't have the contaminant, I think we're heading towards a problem of getting enough to supply the clinical studies that we keep seeing maturing over time. But I'm pretty sure that some companies are taking it into account. Maybe John can weigh in. I know that his company is focused on actinium-225.
Sure, and I'm sure Deborah will have some perspective on this as well. I mean, I hearken back to the early days of lutetium-based therapies, where supply was a huge question. And in fact, if you go back to when I was a graduate student, it was moly supply for technetium. The one thing I'll say about the world's isotope production infrastructure is, as clinical data emerges, the technology evolves to be able to provide the supply. So there's always a small amount of isotope at the beginning as there's, research and development. But as you start to see clinical demand, clinical impact, it grows. And certainly what we're seeing is a number of, the current suppliers are doing a phenomenal job providing material, but we're seeing increased numbers of companies interested in producing in this space. And it really parallels what I think we saw with lutetium, gallium being another, that clinical data drives supply. And increasingly, since we started looking at actinium, we've seen that.
Great.
Yeah, I couldn't agree more, John. The demand will probably fuel inventiveness. And I think our colleagues in the nuclear physics field, who have been developing this actually for quite some time, I think that there'll be more and more routes towards an agent like actinium-225. So I think the supply will be there as the demand evolves.
Great. All right, let's ask a question. Here I have a question from Stephen in the audience. Okay, so Stephen's question goes, for theragnostics, how important is a radioisotope pairing? For example, copper-64 with copper-67 of the same chemical element for the imaging and therapy modalities.
Maybe I can try. I mean, obviously, you want to, the ideal is to have the same element because the electron configuration is going to be the same, and even within the cage, you're going to have the same physical chemical characteristics. What we've learned is that oftentimes gallium and lutetium are just a good pair depending on the tracers. And in the case of PSMA, we even have different ligands. So we have run the vision study with PSMA 11 and gallium labeling as an imaging agent, and PSMA 617 as the therapeutic tracer labeled with lutetium. I think there is always kind of the gold standard that I think copper 64 and 67 may be, but then you also look at the half-life and you look at the range that the isotope has in terms of the energy level. So I don't think there is one size fits all also for the isotopes.
Yeah, I would agree. And I think, Ohad, you talked about this earlier about cross-validation, making sure that different isotopes are giving you the information between the therapeutic and the diagnostic. And there's really the practical component of supply, ability to use those isotopes within the nuclear medicine departments that you're collaborating with. So I think as long as there's the appropriate scientific validation, it gives you the opportunity. But there's no question that using exactly the same structure construct, is kind of the high level gold standard, but there are other ways to achieve the same goal.
Yeah, I totally agree with both Guillermo and John. Having the copper-64, copper-67, or the lead-212, lead-213 combination, it does make your life simpler, for sure. Is it a must? I don't think so. I think if you're comfortable using the current status, for example, with gallium-68, where it's available, it's much more available, and it's easier to handle, for example, for an imaging agent, you should go for it because there's ways around it. And as Guillermo said, it's a nice to have, but we don't even necessarily have the same molecules for the imaging and for the targeting. So having the same isotope, I really don't feel that that's a must.
Yeah, maybe one other quick point is that, again, I'm going to go back to in our field, we've tended to gravitate toward particular isotopes or gravitate towards particular targeting classes of targeting molecules. Each application is unique. And so it's to use, again, the right tool for the right job. In some cases, you'll want PET images. Sometimes you'll want SPECT images. Sometimes you want short half-lives. Sometimes you want longer half-lives for the diagnostic pair. So really, it's pick the right tool for the job and make sure the science supports the use of those particular pairs.
Wonderful. I had this next question, but I feel as though the panel has answered a good part of it here. But here's from David. As ligands and radioligands are developed, what are the theoretical payload limitations? Might one pair an isotope with a known therapeutic or toxic compound for a double approach? But how does this limit differ when this is attempted to cross the blood-brain barrier?
It's a loaded question. There's a lot to unpack over there. I'll give one of it a shot. I think trying to load two isotopes, if I understood the question correctly, trying to load two isotopes on the same molecule, I'm not sure I would go that route. There's a lot of the radiometals used have non-radioactive metals mixed in with them, so cobalt and iron and so forth. So when you're optimizing for the radio labeling with a single isotope, if you want to start mixing the both of them in, or labeling with one and then radio labeling with a second, I think that can be challenging. I think an easier path would be, for example, if you wanted to take PSMA 617, then you can give patients a dose of lutetium labeled PSMA 617, and then follow it up with a actinium radio labeled PSMA 617. And these studies are starting to come out as well. I think that's a simpler path forward than trying to put both isotopes on at once.
And we're probably going to learn much more about patient profiling. So some patients may benefit more from an actinium-based therapy. Conceptually, for instance, when you have micrometastatic disease, some patients may benefit more from a beta therapy, for instance, when you have clonal evolution and heterogeneity in the tumor, where not all cells express the epitope, right? So as we learn more about the practical application, it all comes back to clinical data will rule the day, and we'll see how to best combine and target individual patients with individual tracers, with individual isotopes. And we kind of compare it often to a Lego system, right? You have these building blocks of the target, because we could also consider multiple targets on the same tumor. We have PSMA and GRPR on the prostate cancer cell. Should we combine them? Should we sequence them? When should we use them? We have the ligands, different ligands. Monoclonal antibodies, small molecules exist against PSMA. Should we combine them? Should we sequence them? We have then the isotopes. And I think there's way more research needed to really get the most out of this emerging technology. It's so fascinating to see all these opportunities that I'm very happy to see this renaissance of radioligand therapy emerging now.
That's exactly what I was thinking when I saw the question. Is people are now beginning, and this includes pharmaceutical industry, biotech, as well as academia. People are starting to think creatively about how you can leverage the mechanism of action, how you can combine innovative components of them, and we're just scratching the surface. So it's a pretty exciting future going forward.
Great.
And one more thing I'd like to add is for a lot of therapeutics, we try to get as much as we can into the tumor to destroy as much as we can, as fast as we can. I think one thing that's come up with some recent publications is that sometimes a lower dose of radioactivity can do some interesting things in the tumor. It can maybe lyse a few of the cells, cause neoantigen expression, encourage the T cells to move in, to encourage DCs to start presenting the antigens more effectively. So I think there's really a lot in front of us to figure out. Maybe a lower dose of the radioactivity combined with a checkpoint inhibitor, maybe that'll be a lot better than just trying to demolish everything within the tumor and then damaging the CD8 T cells as well. So yes, there's so much in front of us. It's really exciting.
All right. Well, great question. Thank you for that. From the audience, I'm going to combine two questions now. So Stephen in the audience, I'm going to combine your question with Alan. I'm going to read Stephen's first. I'm going to read Alan's because Alan's is a little bit more direct. But here goes from Stephen. For personalized medicine, is it essential to have a PET isotope with a long enough half-life to image at several time points to do prospective dosimetry for the therapy step? And so, but Alan asks, is dosimetry a must?
I can start with a second one, maybe. Is dosimetry a must? Definitely. I think dosimetry is a requirement for any clinical study that you want to run. I think if you're dosing intravenously and you're dosing enough activity and the decay chain allows you to image the distribution of the agent, then you're definitely going to be expected to image and perform dosimetry studies to make sure that patient safety is maintained. I think beyond that, if you're doing something that's really untraditional, if you are dosing the radioactivity intrathecally, if you're dosing it with direct infusion into the brain tumor, for example, I think it becomes an even more important and challenging question. I think the standard models may not be sufficient to figure out. So if you're infusing something into the brain using the standard models that are expecting something to distribute first within the whole body and only then to get into the brain, they may not be sufficient. So yes, dosimetry is a must. And yes, if you're thinking of doing something that's non-standard as far as the administration pathway, you should seriously think of some advanced analysis incorporated into your clinical studies.
Maybe I can just go ahead, Deborah.
I was just going to chime in on Stephen's question about the half-life, question about the isotope. I think it does depend on the question that's being asked. So if the question that's being asked is, does the patient express the target? That question could be addressed with a short-lived isotope like gallium-68. If the question being asked is about the duration of the retention of your agent in the tumor or in other organs, then I think the long-lived isotope would be the most helpful to try to help predict what might happen with your therapeutic isotope.
Maybe I can also chime in. I would differentiate between clinical development and clinical practice. So for clinical development, I agree dosimetry is a must because we have to understand the area under curve of the biodistribution of our therapeutic tracer. So the nice thing about lutetium is that we can do dosimetry straight off the therapeutic isotope as well. It gets a bit more complicated when you think, for instance, of alpha therapeutics like actinium, that you can't do a direct dosimetry easily. In clinical practice, I would say, is dosimetry a must? Not yet. There are very many factors that influence individual patient response, both on the efficacy and the safety side. And the individualized dosing has disadvantages in terms of practicality, in terms of being able to deliver the therapy to a broad range of patients in what I would say the majority of sites. Very few academic sites today are able to squeeze out those extra 10% of efficacy that you can achieve with the patient individual dosimetry. As we learn more about the patient profile, the genetic makeup of the patient and the tumor, radiosensitivity of tumor and other organs, the genetic background of the patient may also influence the radiation sensitivity of safety target organs like the kidney or the bone marrow. Once we have all that figured out, and once we have easier ways to do patient individualized dosing, whether we still call it dosimetry or not, eventually I think 5 or 10 years from now, we'll come to patient individualized dosing. But frankly, most drugs are not used in a patient individual way for simple practicality reason. And I would say for here and now, getting radioligand therapy adoption triggered, it is important to have a good safety package. And we've seen that with both Lutathera and now with PSMA. And I don't think that in clinical practice, dosimetry is a must-have yet.
We've got about 6 minutes or so left. We'll try to get a couple more questions in. Some great questions here from the audience. And here is a question from Kristen. What is the threshold for a good tumor-restricted target? What numbers do you look for when determining whether to go after a target? Who would like to take that one?
Maybe I can start. I mean, I think one of the things that we've learned is it's not a singular data point. It's not just solely expression. There's the expression number, Deborah pointed out earlier, there's differential expression versus normal tissue and differential accessibility. There's two different components there, kinetics of internalization. So you can absolutely develop sort of an algorithm to narrow down the sort of top list of targets that you'd want to go off, but it wouldn't just be expression. I think there's multiple facets. You've heard discussion about pharmacokinetics as well. So it's a complicated, multifactorial process. The great part about radiopharmaceuticals, again, is the ability to take the diagnostic form and test out your hypotheses and do that starting obviously in preclinical models all the way through tox and into patients. So it's a complicated question that people have been working on for decades.
Any disagreements with that answer? Nope. Okay, I want to ask a question from Michelle. Michelle's got a good question here. Is centralized manufacturing necessary? Or is there a path forward for shorter half-life emitters like Astatine-211, where you may need to establish on-site manufacturing to distribute within a smaller radius of proximal hospitals?
It's a good question. I think you can split it just like we did the question before, like the dosimetry one. For drug development, I think having on-site production, it's easy to set up a few, 2, 3 clinical sites that do have the infrastructure and do have the capabilities to support it. I think if you're looking towards developing the next PSMA 617, centralized production really makes life that much easier. I think if the half-life of the isotope is long enough to enable centralized production and then shipping doses, for example, across the US, I think it'll make an adoption of a therapeutic agent much easier, because trying to produce something for each site, I think it's going to be, it's just, I'm thinking of the CMC headaches of setting up 20, 30, 100 hospitals to produce a therapeutic. It's just, it sounds intimidating to me, but maybe Gilmore, you can weigh in on this, or John.
I mean, having run a radiopharmaceutical manufacturing organization, right now our head of manufacturing is probably going, don't even think about it, because I mean, it's certainly challenging. However, I'm going to go back to the thesis I had before, is there are no absolutes, right? I would say that you have good data and a short half-life diagnostic, people will find a way to sort of get into patients. Gallium being an example. I mean, even FDG, F18, which is manufactured regionally, it's because of the value to the patients in the clinic that drive it. I like centralized manufacturing because this is complicated. This is a complicated process with, specialized requirements. And so I like centralized manufacturing, but if there's great data elsewhere, then people will find a way to do it.
All right. Well, a couple more questions I think we can get in. Let me ask a quick round for the panel over here. A question from Fred. Do you see applications for theranostic approaches beyond oncology? And if so, which indications would be at the top of your list?
Maybe I can get started. In the long run, yes. In the short run, no. Top line answer. I think we have to look at the safety profile, and even more so at the safety perception of any radiation therapy. So when we look at non-malignant disease, the perception is that they're less lethal, even though we have quite a number of diseases that have overall survival well in the range of malignant tumors. The willingness of physicians to use a drug that is conceptually burdened with secondary malignancies, like any form of radiation, that hurdle is high. And I think it's our obligation to provide solid evidence that drugs can be safely and effectively used in non-malignant diseases. And the hit list really goes with the severity and the medical need, the unmet medical need. There are a number of fibrotic diseases, for instance, that have a very poor outcome and not a lot of therapeutic options. Those could potentially be targeted with low-dose radiation or standard radiation radioligand therapies. But I would expect the main focus for the next few years, until we understand much more about how radioligand therapies work, is going to remain in oncology.
Yeah, one thing we unfortunately didn't get time to touch on also is the training aspect and how physicians interact with each other between the different disciplines. That's something that has to evolve and grow as well. And if you want to, I mean, I think the oncology and nuclear medicine and radiology communities are starting to really do a great job coming together, but there's more that's needed. Now, if you bring in another specialization, you have to think even further there. But I agree, short-term oncology, longer term, there are opportunities.
Okay. All right. And that's going to be the last word for today. That's all the time that we have. I want to thank Invicro for sponsoring today's panel. All of our experts today, John Valiant, Guillermo Garicki, Deborah Charych, and Ohad Ilovich, for sharing your knowledge. and everyone in the Endpoints audience for spending some of your time with us today. You can watch your inbox for a link to a replay. And for Endpoints, I'm Arslan Arif, and hope to see you at the next virtual event. Thanks, everyone.
Thank you. Bye-bye.
Thanks, everyone.
Thank you.